सॉरी बात करके टेक्निकल इशू आ जाता है, बिल्कुल ऐसे ही है जैसे बंदा जब जवान होता है तो बहुत सारी चीजें सोचता है लेकिन वो होती नहीं। नेट भी ऐसा ही कनेक्शन है कि बात का तो साथ तो बड़ा अच्छा देता है लेकिन लेटेस्ट टेक्नोलॉजी के साथ साथ प्रॉब्लम्स भी क्रिएट करता है। तो इट हैपेंस नॉर्मली। So just we are going to reconnect. If there will be any issue, so we will start next talk that will be physically. So might be because साढ़े प्राची दा कंप्यूटर लेटेस्ट है ते साडी टेक्नोलॉजी शायद पीछे है ते कोई वजह से प्रॉब्लम है, लेकिन अदरवाइज वी आर गोइंग टू ट्राई to reconnect. Otherwise we are going to start with the permission of reducing with ourselves. So we are going to start after. If there will be this problem continues, we will start next talk. Thank you. Thank you for your patience. I am feeling elated here by seeing the smiling faces. So nice of you.
नहीं वो इंटरनेट वाईफाई यूएसबी राइट
Apologies for the delay, and I believe we are back, and I hope it stays that way. So I'll continue from here, and so we were talking about alignment-free methods, and the first one that we worked on, that we wrote, was MLDSP, that uses machine learning and signal processing, and it has this pipeline, four-step pipeline. So the first one is going from genomic sequences to something numeric. So for that, we use some numerical representations to map our sequences to numerical series. As you can see, for example, the first one is teaser representation. We just use some numbers and replace all these letters by those numbers. Similarly, we have pure inference means where we use one for one kind of basis and -1 for another, and then just using single nucleotide, for example, just add an M, so just A, we replace all A's by 1 and treat all other 3 letters the same as 0. Let's just right. So coming back, the second step is once we have those numerical series, just transform them using Fourier transform. So it's because it's much faster to work in a frequency domain rather than in time domain. So we exploited that concept from digital signal processing, and instead of working with numerical series directly, we use discrete Fourier transform because those numerical series, they are nothing but just discrete numerical series, and obtain the magnitude spectra out of it. That's just absolute value of your result from Fourier transform. And then we applied Pearson correlation coefficient as a distance measure between those magnitude spectra to obtain a pairwise distance matrix. So if you have n sequences, you are going to have n by n matrix, distance matrix with diagonal 0, and all other ij pairs will give you the distance between I and j, the genomic distance between any two sequences. And then we created feature vectors out of those distances, and those feature vectors are fed to machine learning algorithms for training purposes. So any sequences that we encounter in the future that we can use to predict for predicting its label with our trained models. And as byproducts, so we can generate phylogenetic trees or 3D plots that we call molecular distance maps. This is how this process looks like. The first we have sequences. Then from those sequences, we go from these sequences to numerical series. Here we are just showing them as digital signals, 1D signals. In the second paper, we produced something called chaos-game representation plots. That's just two-dimensional numerical representation of your genomic sequences. And you can see a kind of beautiful pattern over here. So this image is from human genome, and this is you can see the repetitive fractal pattern that's present throughout this figure. And this is the visualization of magnitude spectra. This is how your distance metrics look like, and this is visualization, 3D visualization of your distances. And you can create a dendrogram of phylogenetic tree to study evolution or sequence closeness. So you can produce these trees and analyze them there. And then you have machine learning models that can classify different classes. First paper used around 8,000 complete mitochondrial genomes, everything available at NCBI, which is the largest repository for any kind of genomes. So these were what we had at the moment when we did the study. And then we also tested on can we work at subspecies level, and we tried to classify different dengue subtypes. Then we used a few benchmark data sets of influenza and mammalian sequences, and we did that classification. We did that test at I would say almost all the levels of taxonomy, starting from the very highest level, drilling down to the lowest. So at every stage, we picked the largest cluster and drilled down into subclasses, and we obtained really nice accuracies. So at par or even at sometimes exceeding what we can expect from alignment-based methods. And it worked out really well, and it can also give you some insights into missing information. For example, the figure we are looking at here, when we're looking at family to genus level, you can see some overlap between magenta and yellow. And when we look back at the literature, we then plotted just these and red and blue as well. And when we plotted these red and blues by themselves, we were able to even separate those. And the reason being, there was a paper in 1970 that claimed that they should share the same subfamily, and that was never explored later. And that is evident from our result that these two kind of sequences, they must share the same subfamily, because sequence similarity is so close that once they are placed at one level above, they will cluster together. And this is comparison with another, the first one is another state-of-art alignment tree method, BNC. Most of you must have using it, muscle and cluster W. These are most widely used alignment-based methods. And the third one is, the last one is ARTS, and you can see even we can make a separation where alignment cannot. And this is the benchmark data set. That is nothing we created, but that is kind of standard in the field. And when we plotted our result against the existing state-of-the-art method, and you can see we have clear separation of all the subtypes, even where the other methods fail. And in terms of time, when we complete, so I would like to compare it with muscle and cluster W, which are alignment-based methods, and by default, they do 7 iterations to improve their results. They couldn't finish their single iteration for our largest data set in comparatively 2 hours and 6 hours. And we can do that thing in what's written here, 28 seconds. So that's the time improvement that you get from these kind of methods. So that was significant. And the method was even recommended by the International Society responsible for naming all the viruses on our planet, that we should switch to these methods if we want to keep ourselves up to date with the data that we are producing. And then this is just accuracy comparison and time comparison, which you same thing visually. So after we did that, then we thought, can we improve on this method and can we make it more user-friendly? So we asked the stakeholders, mainly the people from genomics and biology, and they said we'll need something that is standalone. Why standalone? Because in most of the countries, you will encounter an issue of data privacy. Most of people from genomics, they are not comfortable in uploading their patients' data over a server, so they need to work on something on their local machine. They want something like something with a very easy user interface, and that is license-free. They don't have to register. They don't have to do anything. So we introduced some additional features in here. We introduced inclusion of a few more distance layers, including and Manhattan, including, by the way, it's not good at all for genomics because it's going to mess up. And again, if you want to know why, you can get in touch with me. But this is something another, I'd say, side topic. And this is something called cache game representation. So this actually representation proves that there exists genomic signature. So our DNA, it has one repetitive pattern that is pervasive along the genome. And even if you graph partial genome, you most likely you'll find that pattern. And you can see these are different chunks from, I would say, segments of chromosome 1. These are totally random, non-overlapping chunks. And when we plotted 10,000 chunk, another chunk of 30,000 length, another of 100,000, you can see we are looking at the same pattern visually. The gradient is a bit different, the color, just because we are using more lengthy sequences, so there will be more dots, more frequency points. How we go from these numbers to this beautiful figure. So we start with empty squares, this label corners, ACGB. We start from mid and say first letter is C. So we'll plot a dot between the mid of the square and the corner label C. Then we take another letter, and that will give you like previously plotted dot and the corner label G, and the midpoint will plot another dot, and eventually you'll see that the pattern will start emerging. So at the end you'll have kind of beautiful plot. This is how this tool looks like that we created, or I should say I created. So you can upload your own data set, or you can select one of the existing one. You can then choose all the parameters. It will tell you about the progress, about the data set, and then you have all these things. This is a 3D plot where you can explore. You can click on a point and look for what data point it corresponds to, and look for more information related to it. You can look at different composition of different, I would say, subsequences or k-merts or subwords. Then you can look at the accuracy of machine learning algorithms on your right-hand panel. The confusion matrix here tells you true label versus predictor labels and classification accuracy scores. And in the top, the bottom right, you can you have the option of selecting testing your new sequences, which is not part of this data set. So this tool is being used by the University of Western Ontario in their second year genomics and biochemistry course for their assignment and for exploring any different new projects for sequence comparison. Then we also tested this on few other data sets. The details are available in the papers. And the third one, the study that we did on COVID-19, SARS-CoV-2 virus that causes COVID-19 disease, and it was done right when the outbreak started, when nobody was working on it. So the first genome, it was made public by the Chinese scientists around, I would say, 9th or 10th of January. And we worked day and night, sleepless nights. I would say we didn't sleep for a week. And we made the preprint available on 21st of January. So we really rushed this work because we saw scope. There was like apart from China, only a couple of countries were seeing 1 or 2 cases here and there. So it wasn't declared pandemic. Even the virus in our preprint, the first one we released, there was no name of the virus. Nobody knew from where it came from. And as we were using supervised machine learning, and most of the time people will question, okay, you need deep learning if you want to explore the labels of new things, because as supervised, you need to have labels. So how you will deal with when something new is encountered and you do not know where it is coming from. So we thought we thought of it as an opportunity that we have a real world case where something new is exposed to us and nobody knew where it came from. And for people from biochemistry, they'll know that it's, how challenging it is. You will have to sequence it, then you will have to say, look at, even if you want to compare it with alignment-based methods, you need so many reference genomes to compare it with, and you'll compare it with first, you'll say, okay, no, it's different, then you'll compare it with second. Then you look at gene levels, compare them, search them in gene database, and a lot of clinical work, lot of clinical effort. And what we did was, we figured out what this virus is within few seconds. So we were among the very first few in the world, and this was covered by the international media across all countries. So we got a lot of PR because of this paper for sure. When we first published our first preprint, there were only 11 sequences available. By the time it went through the peer review process and got finally published in May in PLOS ONE journal, at the time of that final submission, there were only 29 sequences published across the globe. And now it has crossed more than 8 million sequences, and you can see how fast the data is growing. So what we did, how we exploited the supervised machine learning to predict the qualities of something new, something unknown. So we thought we already know the known viruses, we already have the data. So let's use the existing one as training set, and let's predict the level of this new thing at different levels and see what we come up with. So we started with the same process of MLDSP that I gave you before, but we started at the virus realm level, and then we went to its family and then subfamily, and then so on up to genera. And we were figured out it's definitely a coronavirus. And within coronavirus, it's mostly closely related to sarbecovirus, from where the other previous outbreaks, SARS and your MERV2, they came from the same family of viruses. And we also did a testing with the existing known things, and we figured out the closest sequence and why we need the closest known sequence, because that can give us answers from where this virus came. And we figured that out in January 2020, that the closest one comes from bat. Still till date is the closest sequence anybody has found to this, the very first few genomes of COVID-19. So most likely it came from bat. So that's another 2 sequences, and this is just a visual representation of the viruses, existing ones, and other related viruses known to us. And you can see even the similarity in these figures, they have kind of similar patterns. So the concept of genomic signature got proven again here, And numerically, they are slightly different, and that's enough for us to classify these things. So you can see the red ones are all 29 new code sequences that were really close. It kind of looked at that it diverged from this, it came from this RAPG13. Later, the same was proved and claimed even using alignment by the Chinese people, the lab. In the city where this thing came from, or where this thing was found first, I should say. And then again, look at the time we did it. We compared it around 5,000 complete genomes, and the total time at I would say the highest level when we are comparing against all virus species we knew as human, we just took 323 seconds. And when we are looking at the closest neighbors, we just did that in 2 seconds. Yeah, so that's when, that was the time after this paper that International Society, who even named this virus later SARS-CoV-2, and who named all other viruses, be it dengue or anything else, they mentioned us in their report by naming our method, and appreciated the work we did. And they asked the community, because nowadays we are producing sequencing that at so rapid rate that if we keep on using the traditional methods, we can't keep the same pace. So every time will be delayed. So the things we figured out now, in 4 months we'll say, okay, this is not relevant anymore. And same thing with taxonomy. As I said, the first taxonomy and most of field of taxonomy, it was established based on the morphological characters and not on genomic similarities. And nowadays, like taxonomy of bacteria, it changes after every 6 months, because we know that the names we established in the past, they are not correct. So again, just to keep up that pace and to keep us updated, it's good that we adapt the new technologies that are available across. So our observations are these methods are really good, and it doesn't matter if you are comparing different parts of the genome, you'll still find the patterns, genomic signature, and you can compare it to. Another good thing is it's just basically, at the end of the day, it's just pattern analysis. So we have adapted this to compare text from different authors. So for example, say in literature, you can have a question whether Shakespeare wrote this piece of art or not. So what you can do is just take the proven articles where you know this piece was definitely written by Shakespeare, this was definitely written by somebody else, and then you can say that, okay, my method says that it's probability, whatever, 99% is written by Shakespeare or not. So same thing, we are using this pattern analysis for many other fields. And then we are also using it for establishing genotyping and phenotyping relationship. We are doing disease classification, cancer classification, and many other neurodegenerative diseases. We are also working at studying from where the mitochondria came, because first there was no mitochondria, just nuclear genomes, and what was the origin of it? Because it's again a very debatable topic. We are establishing we are studying the evolution of COVID-19. We did a study based on Ontario that is under review and should be published in a month or so. So there are many interesting projects. We also studying the same, using the same method, ECG data, EEG data. That means data from the patients, the actual patients, because they are, at the end of the day, they are patterns. So you can think of any signals, any numerical series as 1D signals, 2D signals. You can compare even signals from different sensors. So it doesn't matter. Your tool is ready that can compare different signals very efficiently, and we can work even at subspecies level. So even if, say, for example, these viruses, at COVID-19, we have now one paper under review where we can do even, we can compare the different subtypes or different clades of this COVID-19 virus or SARS-CoV-2. Again, so I think the connection dropped again, but so I can't show you the slide. I'll see if it comes up quickly. So I'd just like to acknowledge my team. So this work was done. So I work at University of Prince Edward Island now, and but this was done in collaboration with University of Waterloo, Canada, University of Western Ontario, Canada, the Harvard University, USA, and the databases we used was GISAID, VirusForce, BP, and NCBI, and this was funded by the NSER. And that will be all from my side. So I don't know if we have time for any questions. I can take maybe a couple of quickly. The time allows. Otherwise, you can get in touch with me later through emails or anything online, and I'd be happy to talk more. So thank you so much.
Thank you, sir. Dr. Wasim Heather is Assistant Professor at Homestead University, Islamabad. I would invite him to be here and brief us about cancer classification using ML NGS-based gene expression data.
Thank you so much, David, for inviting me. Professor Usman Sahab, Vice Chancellor Sahab, really wonderful talk, mashallah, outstanding work. My work is mainly targeted at student audience, so they told me mostly these are students. Like, yes, how many? Yeah, I'm students too, because initially we were working on my own domain was NGS data analysis, and then we were asked our quiz course by higher ups to apply machine learning, because that's the buzzword nowadays, right? So when we started our career, the buzzword was bioinformatics. वो कप्तान आपके जो कहते हैं ना touch, from bioinformatics का touch शामिल किया. When we did our PhD, sir, तो वहाँ पे it was like NGS touch, right? And then nowadays it's ML touch. So let's see what ML is about. And before we start, very screen पे practical thing. And can you give me a pointer so that I'll explain it to बच्चों थक तो नहीं गए आप आगे आ जाइए जो चेयर अवेलेबल है बैठे आगे आके कभी देर से करिए पॉइंटर है वहां से नहीं वो मुझे थोड़ा देर से पॉइंट आउट भी करना था बेसिकली दिस इज अबाउट मी आई एम वसीम हैदर PhD American way of writing doctor sir they don't say it they just go with your name I'm in computational biology and bioinformatics group we call it CBBG Department of Biosciences CUI Comstats University Islamabad and Nexen Solutions basically are humble effort to do some trainings about this data analysis and do something good for the community. We'll talk about that in a couple of slides by the end of my talk. Partially, I worked with the Miller Center for Genome Research. They had those state-of-the-art NGS machines, so initially they need help. Somebody's help to train their personnel how to do this data analysis thing. So yeah, next. This is me, and I'm also alumni of this university as well. Initially started with master's degree in biological sciences, MSc from Pioneer University in 97-98. MPhil Biochemistry and Molecular Biology under the kind supervision of Professor Dr. Bushra Mirza. And then I came to ComStats. And after 5 years of teaching, and then when I came to ComStats, they asked me to teach bioinformatics courses. I knew nothing about it. So then I did a master's in computer science from Brani Institute of IT. So I also have, I'm honored to be here at my alma mater. I thank Javed for that. It's a big day for me to speak here. And though I have never been here at this place, when we came here applying for agriculture degrees long ago, it was a college, and यहाँ पे सारे care होते थे. But very nice, mashallah. I'm very impressed. That's a tradition in American universities, I think also Canadian and European universities, when you do your PhD, you sit in front of your professor. Chuni si dalte hain aapke upar, pana sahab, they call it hold, right? So that's a lovely tradition. Next is next slide. ComScience, you must be wondering what is ComScience. So ComScience stands for Commission on Science and Technology for Sustainable Development of the South. As the name indicates, mostly it targets the Sustainable Development Goals (SDGs), focusing mainly on developing countries, and most of the Southern Hemisphere, the countries are developing, right? Next. Yeah, Southern Hemisphere, some of the countries. So Pakistan is above equator. That was actually the vision of Professor Dr. Abdul Salam Saab to have an institute where Pakistan should lead, and rest of the countries should also. And Sir Sahi was also there as my senior for 1 year, pro-vice chancellor. Next. These are some of the countries that are there. The headquarters is in Constitution Avenue, Red Zone, Islamabad. Next. Right. So ComScience established an IT institute, Center of Excellence in IT. Initially, we started as CIIT, transformed in 2018 into CUI, ComScience University, Islamabad. Next. Again. Mouse. Okay. These are our campuses, 7 physical and 1 virtual. Next. My department, a beautiful building, Bioscience building, and these are different themes on which we are working. So my theme is Bioinformatics. Next. This is our core Bioinformatics faculty. 5 of us from USP, PhD from USA. We have people, mostly there are international PhDs and postdocs. Next. Yeah, Bioinformatics, as Gurpreet has also indicated, initially when we started, I said, we have to put the touch of bioinformatics in our work, in our degree. So initially, when we started, it was these things, mainly sequence alignment, protein structures, multiple sequence alignment, pairwise sequence alignment, phylogenetics, those things were there. Can you go on next screen? Okay. And yeah, but when we went to our PhDs, then one side we had this genomic sequencing technologies coming in, NGS, etc. At the same time, half of the people, or most of them, at that time, they were working in proteomics domains. Proteins, they came first. When you go enter into the cell, you come across the proteins. So initially it was proteomics, and obviously there's a big portion of people, money, funding resources in proteomics domain. But then they started thinking of going into the genomics domain. In early year 2000, after this inception of Human Genome Project. Next. Next. So most of the word you will hear in this talk is next, next, next. So let's talk about next generation sequencing, or NGS. You know about Sanger sequencing, okay? So that was when they got this NGS. Why they got it as next generation sequencing? Because in early year 2000, when this century came, or even millennium changes, 21st century, this next generation word was the buzzword out there. Next generation mobiles, next generation airplanes, right? So next generation sequencing. So catchy term. That's why people are here listening about it. Is used to describe these number of modern day sequencing platforms. The companies are Illumina, next, Roche, and some official scientific. That's the technology got famous as Point. And then we have next. Applied Biosystem, they came out with solid sequencing. So these are the next generation sequencing technologies. Our second generation. So when you got something next, what is before that? Previous. But nobody dared to call it previous, because Sanger's method is a gold standard method, so nobody can disrespect his work, and So they thought, okay, let's start it now second generation and call the previous one as not previous, but first generation of sequences, right? So first generation, second generation, and now we are having third generation. Soon we will be having fourth generation of sequences. Next. NGS platforms, abbreviated as NGS, they perform massively parallel sequencing, during which millions of fragments from a single sample are sequenced all together. So you break the fragments, you break the chunks of the DNA. Next, take a picture. So these are, for example, your startup material. So you have the DNA, you break it into small fragments. So when you break it into small fragments, lot of fragments are around, and somebody looked at it, fire of a shotgun. Next. Right? So shotgun, anyone knows? That has a cartridge in it, cartouche of them, and then it has cartridges, chale se hote hain, they hit the innocent birds. So somebody looked at a fire of a shotgun, so he called it as shotgun method. So whatever that method is, finally you end up having huge number of fragments from a big chunk of the DNA, chromosomes or whatever, and then you sequence that into those sequencing machines. Next. These are the instruments in those machines in which you sequence them. Illumina has a process, a slide-like thing. Look at the technology, that's a small slide, and you are sequencing the whole genome on that. The next technology, next. Solid came out with flow chip. Next. Yeah, 454. Roche company's project number was 454. So this technology got famous as 454 technology also. So Roche was the name of the company, and the number of the project was 454. So technology was not famous as 454. So 454 goes with these picotype plates, PTPs. Next. And oil current goes with oil semiconductor chips, the chips that are used in your digital camera. So whatever these instruments are, finally they end up producing a huge number of sequence fragments, which are then reads. And there are how many of them? Billions of them, right? And next, your objective is to, yeah, these are some of those instruments are sequences, they look like this. Next. This one is MySeq from Illumina, and it's there in our town. You have a few places in Rawalpindi and Islamabad. MySeq is there all across the country, I guess. Around 10, 12 MySeq machines are here, right? So the party is on. Yeah, very right. Next. Okay, so gathering around sequencers and not producing something is rather than that, you should have one big center and pool up your resources, and that should be productive. So applications of NGS. So students, this is a cell, right? And this is a nucleus in between, and you have that DNA. If you are capturing the DNA or genome, this thing is called as genomics. DNA makes RNA, so that RNA thing is called transcript, right? Set of transcripts is transcriptome, and the field is transcriptomics. So genomics, transcriptomics. There is an entity in between that's called as microRNA. So that interacts with this transcriptome, this RNA, and interferes with its function. Sometimes it's good guy, sometimes it's bad guy. Okay, so that field is also important. It's called as microRNA. And the field related to this here is genomics. Epigenomics deals with the modifications on the genome, the DNA, right? So you get, your target is to capture the DNA and how its function is modified with these proteins or some other modifications like methylations and acetylations and etc. So genomics, epigenomics, genomics, transcriptomics, they say we are living in omics era, right? Omics. Next. This is the complexity here. So while in omics era, these are beautiful lines from Charles Dickens' Tale of Two Cities. Probably he traveled from France to London. So on one side there is development, on the other side I think it's World War II time. Everything is broken, and you also have the same feelings. We had the same feelings when we were in US. In Pakistan we had earthquakes, floods, right? At that time, we had wave of those terrorism as well. So your feelings are like that. So he's comparing two cities, and he says it was the best of time on one side. It was the worst of time, right? But students, what is that?
It was the age, the size of the case.
Which are the right? So you see there is some repetition here, and there is a message here, and there are commas here, and there are full stops here. But when you look this thing in a DNA shape, next slide, looks like this, right? So there are commas and full stops. They are also in the shape of those letters, alphabets. Next, when you give it to the sequencer, this thing becomes this, right? So book of life, And that has letters in it. ADGCs, 4 letters. How many words are possible? Infinite, 4 raised to power infinity, right? So you need to get these strings which are sequenced and put them back and get that message. So obviously that's not an easy task. Next part, here is the solution. We have Cluster Henry doing that for us. Initially, he was going with some basic techniques, classical methods, which he has mentioned. And now there is an era of artificial intelligence, machine learning. Next. The gene expression studies. So we sequence the fragments. Now we do those alignments of those fragments, and we do those alignments either on gene models or we do that on transcripts. And then we count them, how many copies of each transcript is there. So that is the gene expression. Right? So there is a gene A here. And those are the fragments which are sequenced in the sequencer. So how many copies of those green arrows, that arrow indicates the fragment which is sequenced. In NGS language, and I think teachers are covering this course, you guys are nodding in yes, and some of you are enjoying this talk. We call them reads when they're sequenced, right? What is being the So gene A has how many copies? Green lines, D की कितनी है, C की कितनी है? So we sequence them and then we do that with finest aligners. By the way, blast will not work here. So this is a small short fragment. So we want this to be 100% matched, right? So full match, pattern finding job. Can I say approximate match because some of the mismatches are there, right? Okay, so you count these with some scripts, with some counters, next. And finally you end up here. Now your file is our count file, count matrix, numbers, right? And here in this example, we have the genes on the rows. So each row is a gene, each column is a sample. These first 3 yellow ones are the normal controls, for example. So we go for 6 people here. On the other side, these are cancerous individuals, cancerous cells. These may be in your study, these may be 6 plants, 3 controls, and 3 are treated with something. So treatment group versus control group, right? So we know that in gene expression studies, we need replicates, right, to do a statistical testing, right? So that's what it is. So here our files are, and finally after this, we capture the expression with this technique. This thing is called as RNA-seq. Seek, अच्छा omics का word भी है और seek का word भी है. So everywhere it's seek. Exome-seq, genome-seq, RNA-seq, right? RNA-seq got famous. Before that, क्या technique थी इससे पहले? अभी भी है, still that's existing to capture the gene expression. 1, 2 genes, qPCR क्या हो गया है? मेरी तरफ क्या देख रहे हो? quiz रखते हैं मेरे ख्याल, right? So qPCR or qRT-PCR, that's a gold standard, and thousands of the genes, hundreds of the genes, microarray was there, right? RNA-seq captures the whole transcriptome. Okay, so we cannot go for targeted छोटे region के लिए, that's more expensive. So we can have, so that means a lot of RNA-seq data is produced all over the world. That's there in the databases. Next. Right, so now soon we will be depending upon what Gurpreet has highlighted in his wonderful talk, machine learning, right? AI, artificial intelligence. Before we talk about artificial intelligence, what is real intelligence? Few years back, you're a baby, parents took you to a park and they told you, son, this is a bird, ये चिड़िया है, right? And then you had some distribution in your mind कि इसके ये 2 पर हैं, ये 2 इसके बाजू हैं, इसके ऊपर ये क्या होते हैं, feathers होते हैं, खम्भ होते हैं, and so on. So आपके दिमाग में मुख्तलिफ features उसके आप सेव करते कि आप, then you saw another bird, then you saw another birds, and so then you start distinguishing between the crow and the sparrow and the parrot, right? And then you start distinguishing between the birds and the animals and fishes and whatever, blah blah blah. So that was the real intelligence. So here we are also trying to simulate these things on these systems, on these computers, right? So artificial intelligence, born after a conference in Dartmouth, USA, that's what I got it in literature, 1956, and their idea was to, mimic make some machines and mimic human actions. Right? So they were trying to use recently developed computers to simulate various aspects of human intelligence. Next, right types, VAI. While we were studying AI with Dr. Jamil Sawar at Barani Institute of IT, we were thinking to have some computers that should be talking to us, and we were typing something to the computers and set up responses we already recorded in the codes, and they were replying to us, and that was really funny, and that was really interesting for us. So that thing was, now we call it as VKI. So we were having talking computers, right? Suggesting you something. Strong AI, the idea was to develop something like next. This thing, who remembers that? हमारी generation, Purvi ji, मैं वही बड़ी जल्दी कर दी थी बचपन में, तो फिर वो शौक हो गया कि चलो अमेरिका जाना किसी बीच, और आपने पूरा करता. So anyways, Knight Rider, there is a car that talks to that gentleman Michael, याद है? रसोना सा मुड़ा होता थी. वो जैसे बाद में बेवाच में भी काम किया था. Next, so talking cars, they are there in the market right now. Tesla, self-driven cars. You can sleep and they can drive it for you, and there's a car around. आगे को पुलिस वाला खड़ा है, so it will wake you up. Right? Otherwise, he will give you a ticket. So next, machine learning is the branch of artificial intelligence. Within machine learning, we have different types. Rupriji has mentioned in his talk. Again, so just बड़ा बता देता हूँ. Unsupervised, you just leave it to the system, right? एक बच्चे को आप कुछ चीजें सामने रखते हैं और ask him to separate into different things. So for example, मेरे सामने ये जावेद भाई के सामने जो चीजें पड़ी हैं, तो I will put the papers on one side, the water bottles on another side. So this way I can classify the things on myself, without any help or training, without him telling, okay, this was a bird, this is a sparrow, or something like that. If you are having that sort of training, it becomes supervised learning, right? And then obviously there is reinforcement. There is some reinforcement, right, with some reward thing. Next. Okay, that can be used everywhere, and I think Gurpreet ji has covered each and everything, so I don't need to go into that slide. Next. Right, our work was just, again, students are our force, our power, right? So since we were supposed to do this thing and put a touch of ML in our research, so I asked one of my students, Noman, and he picked it up. I said, why don't you take up your gene expression data over the internet? And he went and he found a lovely data on TCGA, the Cancer Genome Atlas. So that has, I think, 21 cancers. Lot of samples, around 100,000 samples, they were there, and we said, let's try these ML techniques and classify these cancers on the basis of this ML thing, right? Classification, यानी एक तो binary classification कर दो. Binary means के distinguish between normal controls and cancers, and then is multi में different types of cancers, how can we separate them out? Or by the way, this is very beginner's work or naive work, and still we can proceed further. All right. So classification of tumors from adjacent nerve samples, binary classification, as well as multi-classification of tumors from the tumor types. Next. And then we did comparative analysis of various machine learning techniques and see how they perform in comparison to each other. Next. This was his main workflow. That's how he worked. Next. Again, that classification example, since we also use classification and we also use the unsupervised, I don't know, supervised media or unsupervised media. In supervised, you have main two big entities. One of them is classification. You classify into different classes, and then the other thing is prediction or regressions as well, right? Regressions तो सबने पढ़ी हुई है ना? तो आप machine learning पढ़ रहे हैं, by the way. And if you are using these PCAs and those hierarchical clusters and dimensional scaling plots in your work, so that's unsupervised machine learning. So we're already doing that. Trust me. अच्छा, यहाँ पे we got another undergrads project, so in which she had this data. So we have the data of this gestational diabetes. Just to make you an example. 6 pregnancies, glucose 148, blood pressure 72, skin thickness 35, insulin 0, BMI is this, diabetes pedigree function, pedigree में है नहीं है, strong indication, and then age is 50. What do you think? What do you think? 1 means diabetic, 0 means not diabetic, right? So first we give this way, we give the file to the system in this fashion, we put ones and zeros. So this all ones and zeros are your labels. Next, what if I remove those labels? Will you be able to distinguish with your real intelligence? Of course, you can do that. So if you are given like 100 rows, maybe you can go like 70% accuracy and you can get those patterns very well and distinguish between zeros and ones. So this thing is the labeling which we are doing. So this is the real data. We have those cancers, normals and controls. So actually we transform the files, Python code, very simple and easy. Even, people in humanities are doing that. Luigi has also mentioned this language processing, right? So even people doing with masters in English, they are doing these things. So students, you can do it. Next. So we have these labels, we transform the files. Now our columns go to rows and rows go to columns, and finally we can put those labels. Zeros are normal, controls, ones are cancerous, and then this part is we give it to the system. When we give it to the system, he learns from it. So that's his training, learning. So that's where he's learning. Next, what we do is testing. In testing, we remove the labels. So that was his main method. Right, so we tested these models. Some of them are here. SVM, ये सारे मुख्तलिफ machine learning methods हैं. हमने कहा देखते हैं इनमें से कौन कितना अच्छा predict करता है. ठीक है जी. 21 cancers, around 1000 samples. Next, we divided the data set into 70 and 30 based upon the literature. I would like Gurpreet to have comments on this as well. 70-30, I think that's okay. 70% testing and 30% training. Next. And then various functions are not going into it, and various matrices next, which we use. अच्छा ये हमारे मुख्तलिफ cancer types हैं और उनके कितने कितने samples हैं, how many normal controls, how many cancerous patients were there. Next. Yeah, again their distribution. Next. In unsupervised machine learning, as I said, we leave it to the system. So you see those blue dots and you see those red dots. The red ones are normal, blue dots are different types of cancers. Do you see their separation from each other? Do you see those red dots separate from the blue ones, or at least not mixing in between them, right? So that means this thing is working. There is something different as far as the gene expression is concerned. Why to focus on RNA? Why not to focus on proteins? Because RNA, they come first. Normally, the present diagnostics is all protein based, and then we go at, sometimes we detect it at T4 stage, T3 stage, right? So it's too late. So they say if we focus on the RNA things, then we start developing the things that can check your gene expression within your body. So we can do some safety measures, right? Next. Again, multi-class maybe, so we get this unsupervised machine learning give us the results like this. So PCAs are dimensional scaling plots, you must be familiar with them. They just separate those things. So dots, since dots are the same colors, same cancer type are together, so they are coherent. Dots of the different colors are separate, right? So there is separation. Next. Asha ji, I want you to understand this picture. How many of you have done stats courses? False positive, false negative, pata hai? True positive, true negative. There are 4 possibilities. True, false, positive, negative, right? So, because of the cultural barriers, I will not like to explain this thing, but you can get the pattern by yourself. So 1, and he picks it as 1, so that's true positive. 0 picked up as 1, so that's yeah, false positive, right? And then false negative. 0 was picked up as 1. And 0 was picked up at 0. What thing you like the most in the idea of this? Obviously, we like the truth, be positive or negative. True hona chahiye. Within that, maybe most of us are biased towards true positive. So true positive hona chahiye, true negative. And what you hate the most? What should not be there? Negatives nahi hone chahiye. Sorry, false nahi hone chahiye. Right? False चीज nahi hone chahiye. Negative or positive, right? So you put them together, so you get those equations: accuracy, precision, and recall. And that's what we calculated for these algorithms. Next. Our results for logistic regressions, that's what we got in this matrix. Probably she is coming here to one. Next. Next, and you guys are anxiously waiting for tea, I know. So these are accuracies of these various are medalists. So most of them, they were like 90 or 99%. So everything is working here. That means there's a big difference in gene expression of normal controls versus cancers. Different types of cancers even. So with gene expression data, you can detect this thing, right? Next. These are area under the curve, area under the receiver operator curve. Receiver operator, because they were working with radars initially to distinguish between the signal coming from a real aeroplane and a bird, right? So that's where they developed. So in machine learning thing, we are actually focusing on these things a lot. इसमें area under the curve देखना है. So कौन से curve में सबसे ज़्यादा under the curve area है? Red वाले में, green वाले में, या फिर शुरू वालों में? Which one is the good curve you think, and which one is the bad curve? Based on the area, right? वो बिल्कुल जाके आगे लोकेट से राइट हो गई. लाहौर वाले कहते हैं टक्कर को सजेल देना, ठोकर को सजेल देना, right? So you go at the end, so मैं आ रहा था, so my guide asked me कि बिल्कुल आखिर में जाएंगे, there is the wall and then you turn right. So those are the ones that are the good curves. The ones which are taking shortcuts, they are not good. So these curves tell us shortcuts are not good. Next. Actually, we also tried these neural network models or deep learning things, and their efficiency was more in multi-classification. Binary में दोनों ने ठीक एक तरह का काम किया है these approaches. Okay, let's conclude. अच्छा ये हमारा confusion matrix है multi-classification की कि कौन सा cancer था, which cancer was there and which he picked it up, right? So most of them are on those diagonals, so they were good ones, and some of them they were a little bit off. And over here we noticed that it was off for the similar cancer types, right? तो एक तरह के tissues के cancer से उनमें जरा mixing आ रही थी. Next. So we conclude the best slide you might see here, the last one. Machine learning can be used correctly to classify cancers. Normal individuals, data set used in this study as no one have implemented using set of numbers and all. That's a big claim. We saw there are a couple of papers. So some of the one paper from India, I think they got 3 or 4 cancer type, and one paper from, of course, China. They got 16. I was asking Noman to publish, and he was the thesis of it. And then after his thesis, he's just gone. He's a sign कर दिया बच्चे का, जवाब आपके college की हो गया. Right. So that is still unpublished, but we have 21 cancers here in this, and you guys can help us if anyone is interested, especially the people in molecular biology, right? Next. Oh, this is our humble effort. So we established a company when I got back from US. So my family, we got my brother, he lost his life as a young army captain, very outstanding engineer in an accident. So we developed, we organized some money and we did some community services. And my sister, she's a medical doctor next, she was the leader of that. So I thought maybe let's organize these trainings. At that time, we started with NGS data analysis things. And then we give the money to the community. In fact, we good about it. That's why I'm here. That's why by your name we got connected. Somebody told him that this guy can speak. Next. Some of our trainings. Next. A glance of our activities, community services. But my father up there passed away of COVID last year. We got bless him. So he was the big supporter of, motivator for us. Thank you so much. Thank you so much. Thanks for your time. So this is our group. Noman, who did this work, is in the first row here, the tallest one, second on your right. Thank you so much. Any questions, concerns, queries? Yeah, chai the I मेरे को तो आप chai का ही पूछेंगे. Thank you so much.
Dear audience, I would again invite Dr. Gurji, as the connection was interrupted previously. So, sir, please come here.
And thank you. So I'll share the slides. This photo देखनी है वैसे. हाँ जी, मैं actually I want to talk about like इथों तुसी केड़ा मैसेज लेके जाना चाहते हो कि अगे तुसी की कर सकते हो, right? So what we did ਅਸੀਂ show ਕੀਤਾ. So ਹੁਣ ਤੁਸੀਂ ਕਿੰਨਾ contribute ਕਰ ਸਕਦੇ ਹੋ ਸਾਡੇ society ਨੂੰ, university ਨੂੰ. When we talk about मैं by profession or by teaching, I'm a data scientist. ठीक है, मैं computer scientist हूँ, so I work with variety of data, different domains. So I started with genomics, biology, and I was new to that field when I went abroad. And ਮੈਨੂੰ ਵੀ biology ਦਾ limited experience ਸੀਗਾ, and I had to learn. And same way, ਹੁਣ ਅੱਜਕੱਲ ਦਾ ਜੋ ਜ਼ਮਾਨਾ ਹੈ, I would say if you want to stay on top, ਤੁਸੀਂ ਸਿਰਫ ਆਪਣੇ field ਤੱਕ limited ਨਹੀਂ ਰਹਿ ਸਕਦੇ. ठीक है, so if you are a computer scientist, you need to learn something new. ਜੇ ਤੁਸੀਂ biology ਹੋ, biochemistry ਹੋ, you need to have other departments. Collaboration, interdisciplinary work. When we work as data scientists, एक data scientist का 70% effort does that data collection and cleaning. That is the most important part. Machine learning components, algorithms are there. All you need to do is put in data, get the results, and then analyze them. That's really simple. Libraries are there, codes are there, examples are there. Main important thing is getting that data. When data depending on your environment. When we talk about health data, when we talk about genomics data, जो आप see databases देख रहे हैं, NCBI, जरा cancer atlas है, इनका तो सारा most of data, I would say more than 90% is based on West. ठीक है, अपने South Asian data is very limited. So you are doing work for West, even though you are contributing to research. I'm not saying that's bad, but what we are doing for our communities over here. ठीक है, so ਸਾਡੇ ਕੋਲ ਇੱਕ ਵਧੀਆ opportunity ਹੈ. You are sitting at a place ਜਿੱਥੇ ਸਾਡਾ agriculture ਦਾ ਇੰਨਾ ਜ਼ਿਆਦਾ data produce ਹੋ ਰਿਹਾ that you can work on. Doesn't matter if ਤੁਸੀਂ computer scientist ਹੋ ਜਾਂ ਤੁਸੀਂ biochemistry ਤੋਂ ਹੋ, ਜੇ ਤੁਸੀਂ computer scientist ਹੋ, so precision agriculture ਤੋਂ sensory data collect ਕਰ ਰਹੇ ਨੇ, ਜਿਹੜਾ crop ਦਾ yield ਦਾ ਬਾਕੀ data collect ਕਰ ਰਹੇ ਨੇ, ਤੁਸੀਂ ਉਹਨੂੰ analyze ਕਰ ਸਕਦੇ ਹੋ, machine learning use ਕਰ ਸਕਦੇ ਹੋ. ਜੇ ਤੁਸੀਂ biochemistry ਤੋਂ ਤੁਸੀਂ interested ਹੋ ਕਿ ਮੈਂ sequencing data ਦੇ ਨਾਲ ਹੀ ਕੰਮ ਕਰਨਾ ਹੈ, ਭਾਵੇਂ chip seq RNA seq data ਹੋ ਗਿਆ, ਸਾਡਾ gene expression ਦਾ ਹੋ ਗਿਆ, ਸਾਡੇ complete genome ਹੋ ਗਏ. So you must be aware already CRISPR-Cas9, मैं फैसलाबाद University Agriculture visit की थी. I was happy to see they are working on that. हाँ जी, so डॉक्टर साहब ने जिस तरह उन्हें सर you have access to one of the world's best sequencing machines here. So तारे कोल एक इतना बढ़िया platform है. CRISPR-Cas9, the people who don't know, it's a so simple thing you can do in your lab. जे अपना genome है, basically how you look like, how you behave, what you do. ताकि ये कोई भी diseases ने ताकि health concern ने अगे next generation कितना भी दिया, everything is in your genome. ठीक है, set of instruction है, nothing else. जीनोम एडिटिंग with Cas9 ਹੁਣ ਆਪਣੇ ਜੀਨ ਦੇ ਵਿੱਚ ਜੇ ਆਪਾਂ ਐਡੀਟਿੰਗ ਕਰਨੀ ਹੈ, ਕੋਈ ਆਪਾਂ ਉਹਦਾ ਪਾਰਟ ਕੱਟਣਾ ਹੈ, ਰਿਪਲੇਸ ਕਰਨਾ ਹੈ, Cas9 is like Caesar. ਠੀਕ ਹੈ, ਐਂਜ਼ਾਈਮ ਆਪਣੇ ਅਟੈਚ ਹੁੰਦੇ ਆ, ਜਿੱਥੋਂ ਮਰਜ਼ੀ ਆਪਾਂ ਕੋਈ ਪਾਰਟ ਕੱਟ ਕੇ ਉਹਨੂੰ ਰਿਪਲੇਸ ਕਰ ਦਿੰਦੇ ਆ. ਠੀਕ ਹੈ, ਜੇ ਹੁਣ ਅਸੀਂ ਅਗਰ ਡਿਜ਼ੀਜ਼ ਦੀ ਗੱਲ ਕਰੀਏ, ਕਿਸੇ ਨੂੰ ਲਕੀਮੀਆ ਦੀ ਟ੍ਰੀਟ ਕਰ ਸਕਦੇ ਹਾਂ, ਬਟ ਉਹ ਲੈਵਲ ਤੇ ਪਹੁੰਚਣ ਲਈ ਸਾਨੂੰ ਮੈਂ ਕਹਿੰਦਾ ਕਿ ਬਹੁਤ ਜ਼ਿਆਦਾ ਮਿਹਨਤ ਦੀ ਲੋੜ ਕਰਨੀ ਪੈਣੀ. ਸੋ ਆਪਾਂ ਜੇ ਆਪਣੇ ਸਕਿੱਲਸ ਨੂੰ ਯੂਟਿਲਾਈਜ਼ ਕਰੀਏ, ਸਾਡੇ ਕੋਲ ਐਗਰੀਕਲਚਰ ਦਾ ਇਹਨਾਂ ਵਧੀਆ ਪਲੇਟਫਾਰਮ ਹੈ. ਉਹਦੇ ਵਿੱਚ ਹੁਣ ਜੇ ਅਸੀਂ ਹੁਣ ਬਾਹਰ ਦੇਖਦੇ ਹਾਂ ਫੀਲਡ ਦੇ ਵਿੱਚ, ਜੇ ਤੁਹਾਨੂੰ ਵਿਜ਼ਿਟ ਕਰਨ ਦਾ ਮੌਕਾ ਮਿਲੇ ਵੈਸਟ ਦੇ ਵਿੱਚ, ਤੁਸੀਂ ਦੇਖੋ ਐਗਰੀਕਲਚਰ ਯੀਲਡ ਇਨਕਰੀਜ਼ ਕਰਨ ਲਈ ਜਿਹੜੇ ਫੀਲਡਸ ਨੇ ਜਿੱਦਾਂ ਚੱਲੀਆਂ ਲੱਗੀਆਂ ਨੇ, ਸਾਰੀਆਂ ਸੇਮ ਲੈਵਲ ਤੇ ਲੱਗੀਆਂ ਹੋਣਗੀਆਂ, ਨਹੀਂ ਕਿ ਉੱਚੇ ਨਹੀਂ ਸਾਡੇ ਨੂੰ ਕਿੱਥੇ ਲੱਗੀ. ਠੀਕ ਹੈ, ਕੀ ਹੈ, ਜੀਨ ਐਡੀਟਿੰਗ ਹੈ. more varieties produce करने हैं, yield increase करना. Fruit size, ठीक है, कोई fruit है, कोई crop है, ये उन्हें स्पोर होना ही लगना, बट जो साइज बढ़ जाएगा ताकि yield ताकि by weight increase होना. ठीक है, so agriculture department I know is working on that. You have access to that data. जो नवी varieties से कम करना चाहते हैं, वो gene editing परफॉर्म करना चाहते हैं, कुछ करना चाहते हैं, you can help them. ठीक है, वो data analyze कर सकते हो, किसी ਉਹਦੇ ਉੱਪਰ ਵਰਕ ਕਰ ਸਕਦੇ ਹੋ, because that would be easier for you as well, because you already have access to that data, and that will be really novel work as well. Second thing, I myself मेरी कोई बैकग्राउंड कंप्यूटर कंप्यूटिंग रही है, I worked in image processing, computer vision, I worked on literature analysis, I like exploring things. Recently I'm also I also started working in precision agriculture with Dr. Ezra Saru. ਅਸੀਂ ਹੁਣ precision agriculture ਵਿੱਚ ਕੰਮ ਕਰ ਰਹੇ ਹਾਂ because language exciting field for sure, not so explored, so there's a lot of scope to do something new, not just say at the Pakistan level or at the South Asian level, I would say at the world level. Data day concern may namely whatever, it's a $1 billion market. अजय भी ये देखा जाए कितनी पॉपुलेशन वर्ल्ड की है बिल्कुल फूड नहीं है एंड स्टैट्स आर हॉरिबल बिकॉज़ काफी वर्ल्ड के विच फार्मिंग ਜਿਹੜੀ ਹੈ ट्रेडिशनल फार्मिंग है ਉਹ प्रॉफिटेबल कितना नहीं रहा, सो पीपल आर मूविंग अवे फ्रॉम फार्मिंग और ਉਹੀ सस्टेन कर रहा है ਜਿਹੜਾ ਨਵੀਆਂ ਟੈਕਨੋਲੋਜੀ ਅਡਾਪਟ ਕਰ ਰਿਹਾ है। सो ਆਪਾਂ ਨੂੰ मतलब इनफ फूड नहीं हैगा वर्ल्ड पॉपुलेशन फीड ਕਰਨ ਲਈ। सो एक meaningful way भी है जब सोसाइटी में contribute कर रहे हो as a researcher, it's exciting, you can get high impact people research work out of it and that solves too many problems। ਸਾਨੂੰ ਕਿਤੇ ਬਾਹਰ ਜਾਣ ਦੀ ਜ਼ਰੂਰਤ ਨਹੀਂ ਹੈ, ਸਾਡੇ ਕੋਲ ਆਪਣੇ ਕੋਲ ਹੀ ਸਾਰੀਆਂ ਚੀਜ਼ਾਂ ਹੈਗੀਆਂ ਨੇ ਜਿੱਥੇ ਤੁਸੀਂ ਇੰਟਰੈਕਟ ਕਰ ਸਕਦੇ ਹੋ, ਡਾਟਾ ਲੈ ਸਕਦੇ ਹੋ and reach out to people, that's what we do। क्योंकि ਜੇ ਆਪਾਂ ਆਪਣੇ ਤੱਕ ਰਹੀਏ ਕਿ ਨਹੀਂ ਉਹ ਮੇਰੇ ਕੋਲ ਆਏਗਾ ठीक है फिर देखांगे ਜਾਂ ਉਹਨੇ ईमेल ਕੀਤੀ ਸਰਸਰੀ ਗੱਲ ਕੀਤੀ बात भूल गए उतना काम नहीं होना, ठीक है। सो अपन इनिशिएटिव लेना पड़ा है, गो रीच आउट टू पीपल, एक जना इंटरेस्टेड नहीं है दूसरे को जाओ, दूसरा नहीं है तीसरे को जाओ, आज मैं ਤੁਹਾਡੇ ਕੋਲ ਕੀ ਡਾਟਾ ਹੈ आप मेरे स्किल्स ने मैं कितना कंट्रीब्यूट कर सकता हूं, दोनों ਦੀ विन सिचुएशन है। सो बिल्ड कोलैबोरेशन, अपने ਜਿਹੜੇ स्टूडेंट ने ਉਹਨਾਂ ਨੂੰ ਵੀ ਮੋਟੀਵੇਟ ਕਰੋ, ठीक है। ਜਿੱਦਾਂ ਇੱਕ ਕੱਲ ਬੜੀ ਇੰਟਰਸਟਿੰਗ ਗੱਲ ਹੋ ਰਹੀ ਸੀ ਕੰਪਿਊਟਰ ਡਿਪਾਰਟਮੈਂਟ ਦੇ ਵਿੱਚ ਇੱਕ ਡਾਕਟਰ ਸਾਹਿਬ ਆਈ ਥਿੰਕ ਰਿਟਾਇਰਡ ਸੀ, ਉਹਨਾਂ ਨੇ ਗੱਲ ਕੀਤੀ ਕਿ सारी जी इतने इनोवेशन की कमी है, कुछ चीज ठीक कितना होगी, ठीक है। इनोवेशन कितों आएगी, अपन क्लासरूम तो स्टार्ट करना पड़ना है। अपन उन्होंने असाइनमेंट बच्चों ने लिखे मैंने कि नॉवल कर रहे हैं, असं उत्थे प्लेजरिज्म चेक करना देखते हैं, किसी तक कोई मैच नहीं होता, वो सिस्टम बिल्ड उतो करना पैना है। जो ਤੁਹਾਡੇ अंदर ਤੁਸੀਂ ਕਿਸੇ ਨੂੰ सुपरवाइज कर रहे हो, जो बच्चा लगे होगा कि आज ही इतना का काम होया सीगा, मैं भी इतना का चुक के देता है, उनको नहीं इतना नहीं होता, ठीक है। कम अप विद जो हुण तक होया ਉਹਦਾ लिटरेचर रिव्यू ਤੇ टाइम स्पेंड करो and let's do move forward कि वो तो ਅੱਗੇ ਕੀ ਕਰਨਾ ਹੈ ਉਹ ਸੋਚੀਏ, ਉਹ ਵਾਲੀ ਚੀਜ਼ ਉਹ ਆਪਾਂ ਨੂੰ ਐਸ ਟੀਚਰ ਉੱਥੋਂ ਕਲਾਸਰੂਮ ਤੋਂ ਬਿਲਡ ਕਰਨੀ ਪੈਣੀ ਹੈ। ਸੋ ਉਹਦੇ ਉੱਤੇ ਵਰਕ ਕਰਨ ਲਈ ਮੈਂ ਤੁਹਾਨੂੰ ਇਹ ਸੁਜੈਸਟ ਕਰਾਂਗੇ ਕੋਲੈਬੋਰੇਸ਼ਨ ਵਧਾਓ। ਯੂ ਬੀ ਐਗਰੀਕਲਚਰ ਯੂਨੀਵਰਸਿਟੀ, ਯੂਰ ਫੋਕਸ ਸ਼ੁਡ ਬੀ ਔਨ getting that data and it's easier for you, not just don't think of it as a service that you are helping somebody, it's good for your own profile too because you have easy access to data। You will do something that hasn't been done on the planet, you will be the first to do it। so उससे ਤੁਹਾਡਾ यूनिवर्सिटी ਦਾ ਨਾਮ ਵੀ ਬਣੇਗਾ, ਦੇਸ਼ ਦਾ ਨਾਮ ਵੀ ਬਣੇਗਾ, ਤੁਹਾਡਾ ਨਾਮ ਵੀ ਬਣੇਗਾ। so that's what I would like to say कि ये वाली चीज ਨੂੰ ਸੋਚੋ, ਥੋੜਾ ਸੇ ਟਾਈਮ ਸਪੈਂਡ ਕਰੋ, ਟੌਪ ਤੋਂ ਬਾਅਦ ਭੁੱਲ ਨਾ ਜਾਓ, ठीक है। मेरे ਨਾਲ ਗੱਲ ਕਰਦੀ ਹੈ ਜਦੋਂ ਮਰਜ਼ੀ I'm available, ਮੇਰੀ ईमेल वगैरह सब कुछ शेयर कर देंगे, I'm open to discuss that anyways। ਤੁਹਾਡੇ ਕੋਲ ਡਾਕਟਰ ਪ੍ਰਭੂ ਨੇ, ਤੁਹਾਡੇ ਕੋਲ ਵੀਸੀ ਸਰ ਨੇ who is helping you, ਤੁਹਾਡੇ डायरेक्टर ने। सो ਆਪਾਂ ਸਿਰਫ ਆਪਣੇ ਇੱਕ ਛੋਟੇ ਜਿਹੇ ਫੀਲਡ ਦੇ ਨੈਰੋ ਡਾਊਨ ਤੇ ਫੋਕਸ ਨਾ ਕਰੀਏ, ਆਪਾਂ ਥੋੜਾ ਬ੍ਰੌਡ ਚੀਜ਼ਾਂ ਨੂੰ ਸੋਚੀਏ, ਇੰਟਰਡਿਸਪਲਿਨਰੀ ਵਰਕ ਦੇ ਉੱਤੇ ਕੰਮ ਕਰੀਏ। ਸੋ ਥੈਂਕ ਯੂ ਸੋ ਮਚ। ਅਜੇ ਆਲਰੇਡੀ ਵੀ ਥੋੜੇ ਲੇਟ ਆ, ਸੋ ਅਪੋਲੋਜਾਈਜ਼ ਫॉर द डिले and let's continue with that, thank you।
ਤੁਹਾਡੀ ਬਹੁਤ ਮਿਹਰਬਾਨੀ ਕਿ ਤੁਸੀਂ ਮੈਨੂੰ ਦਿਖਾਈਆਂ ਨੇ।
ਕੁਝ ਵੀ ਜੇ ਇਹਦੇ ਚੋਂ ਮਤਲਬ ਤੁਹਾਡੇ ਤੋਂ 5% ਵੀ ਕੋਈ ਮੰਨਣਗੇ ਤੇ ਬਹੁਤ ਖੁਸ਼ੀ ਹੋਏਗੀ।
Dear audience, now I am honored to invite our chief guest, Professor Dr. Kamrul Zaman, Vice Chancellor, Field Harisha, and Agriculture University of Sydney. Sir, please.
Our distinguished keynote speakers, guests from Canada, Vice Chancellor, Directors, जो ये बातें मैंने करनी थी वो डॉक्टर सुधीर सिंह ने आपसे कर ली कि ये जो हमारी जो जिम्मेदारी है और रिस्पॉन्सिबिलिटी फीड द पॉपुलेशन,
पाकिस्तान की पॉपुलेशन जो है उसको ही मसला हुआ तो हमने फिर वो जो पुअर कंट्रीज है उनकी भी हेल्प करनी है, वो किस तरह करेंगे। तो ये जो टेक्नोलॉजीज हैं, ये बायोटेक्नोलॉजी है, कृषि एग्रीकल्चर टेक्नोलॉजीज है, तो ये जब तक हम इनको अप्लाई नहीं करेंगे तो ये मसला हल नहीं होगा। वो ट्रेडिशनल फार्मिंग जो है वो उससे तो अब काम नहीं चलेगा। क्लाइमेटिक की वजह से देख लें, और जो अर्बन एरिया बढ़ रहा और जो हमारा एग्रीकल्चर लैंड है वो कम हो रही, क्योंकि वो आप देख रहे हैं कि हाउसिंग सोसाइटीज जो बन रही, क्योंकि पॉपुलेशन ग्रोथ रैपिडली इंक्रीज हो रही। तो ये लेकिन ये जो बायोटेक्नोलॉजी आपका जो फील्ड है, जिस तरह ये डॉक्टर साहब आए हैं कॉन्सेप्ट से, तो ये इन्होंने वो एग्जांपल दी, तो वो हम अगर वही टेक्नोलॉजीज हैं, ये देखिए जो कृषि एग्रीकल्चर का जो मेरा फील्ड है तो ये समझ नहीं आई वैसे, लेकिन वो आर्टिफिशियल इंटेलिजेंस और दूसरा जो वो चीजें जो है ये एग्रीकल्चर वाले पहले कहते थे जी इसका काम क्या है कि जी मैथ्स स्टैट पढ़ ली, वो डॉक्टर नहीं सब बैठे वो उन्होंने पढ़ा दी, पास हो गया, ग्रेड आ गया, बस ठीक नहीं। अब देखें कि ये सारा इसके पीछे मैथ है, स्टैट है, फिर जाके वो कंप्यूटर वाले आते हैं, फिर आप वो और इसको अप्लाई करने के लिए जैसे डॉक्टर सुजीत सिंह ने कहा कि मैंने बायोलॉजी सीखी, डॉक्टर साहब वो हेल्थ साइड पे। तो हमारा जो है ये जो एक्सपेक्टेशन आपसे बहुत हमारी हाई है कि क्योंकि मेरा 27 साल या 28 साल हो गया यूनिवर्सिटी में नहीं, प्रो वाइस चांसलर साहब बैठे, तो कोई एक भी वैरायटी नहीं है हमारी। तो अब जो वो ऑलिव की हुई है, जबकि बाकी लोगों की पता नहीं कितनी कितनी ऑलिव की पहले हो गई, तो ना मीट की है, ना राइस की है, ना वेजिटेबल की है, किसी चीज की कोई वैरायटी नहीं है। तो पेटेंट वो अब फाइल हुए हैं और पेटेंट भी नहीं है। तो लेकिन वो प्लांट ब्रीडिंग जेनेटिक्स के मेरा नहीं ख्याल कि वो कोई भी बैठा है, आए ही देख लो। और बहुत बड़ा मसला कि बायोटेक्नोलॉजी, बायोकेमिस्ट्री, प्लांट ब्रीडिंग जेनेटिक्स, ये सारे और ये इवन एग्रीकल्चर के सारे डिजीज के मतलब जो भी बेशक हेल्थ की है, बेशक प्लांट की है, बेशक एनिमल की है, तो लेकिन देख ले कोई नहीं बैठा, तो इतना इंटरेस्ट है। और लेकिन मैं पेशावर हम गए कैपेसिटी बिल्डिंग और ट्रेनिंग के लिए, तो वो उन्होंने हमें वो बायोटेक्नोलॉजी बताया। उन्होंने डॉक्टर साहब स्पेशली जब साहब के लिए वो हमारे बोर्ड पे भी हैं, वो आते हैं, वो उनकी वैरायटीज हैं भी जिनको हम कहते हैं जी कि पेशावर एग्रीकल्चर यूनिवर्सिटी नाम क्या है, कुछ नहीं। एरड दूसरी मेरा ख्याल सेकंड लार्जेस्ट यूनिवर्सिटी एग्रीकल्चर की पाकिस्तान में और वेल नोन हो गई। लेकिन ये आप जो है ना, ये अब इतने फैकल्टी बायोटेक्नोलॉजी, बायोकेमिस्ट्री, तो अगर नहीं भी ये बीबी वाले आते तो जैसे उनका कि हम उनके पास चले जाएंगे कि आप नहीं, जब तो हम अपना काम शुरू करें तो कोई एक दो चीजें जो है ना वो अब नजर आना शुरू कर। तो मैं कह रहा था कि ये टेक्नोलॉजी जो है, ये जीपीएस जब ये यूएस मिलिट्री वो यूज करते थे, वो कोड देते नहीं थे किसी को, डिफेंस के लिए था। 1970s में वो एग्रीकल्चर को कोड किया, तो वही चीज उन्होंने एग्रीकल्चर में अप्लाई करके फिर वो जीआईएस सॉफ्टवेयर, रिमोट सेंसिंग वहां से शुरू करके अब देखें कि वो कितनी टेक्नोलॉजी एडवांस हुई, ये आर्टिफिशियल इंटेलिजेंस, डीप लर्निंग, मशीन विजन, जो आता ना वो एग्रीकल्चर में अप्लाई कर, सारी मशीनें भी ऑटोमेट हुई और वो डाटा सारा जो है वो रिमोटली कंट्रोल होता। और हमारे बाद ही वो डाटा ड्रिवन कोई चीज है ही, वो सबसे बड़ा चैलेंज ये है। तो अब ये हमने डाटा सेंटर जो बनाया प्रोसीन एग्रीकल्चर का, अच्छा वो जीपीएस जो फिर वो यूज होना शुरू हुआ, फिर टेक्नोलॉजी महंगी होती पहले,
वो मैं जब 95 में गया तो यूरोप में अर्ली 90s में शुरू हुई, तो वो बारो करते थे सर्वे डिपार्टमेंट से, तो वो 7,000 पाउंड का वो बैक वाला जीपीएस, तो वो हमारा सुपरवाइजर का बड़ी ध्यान से भी एक मसला हो गया कि इतने पैसे कहाँ से आएंगे, एक था यूनिवर्सिटी में। तो लेकिन आप जीपीएस देखें, वो आपके फोन में भी है, आपकी गाड़ी में भी है, हर जगह कंप्यूटर जो आप इस साल यूज कर रहे हैं, नेक्स्ट ईयर वो स्लो हो जाएगा। वो हमने देखा 2 साल बाद वो काम कर जाता फास्ट कंप्यूटर टेक्नोलॉजी। अब ये आईफोन हर 6 महीने बाद नई वर्जन आ गई टेक्नोलॉजी, तो और जिस तरह इस्तेमाल शुरू होता है फिर सस्ती होनी शुरू हो जाती है। तो इसी तरह से ये जो आपका जो बायोटेक्नोलॉजी का है, अब वो कहेंगे जी कि एग्रीकल्चर वाले तो हमें तो जरूरत नहीं आती, आप जाके तो अपना टेक्नोलॉजी करें। लेकिन हालांकि ये जितनी एप्लीकेशन इसकी जो मैंने देखा जितना वो एग्रीकल्चर में है कि अब वो हमने एक और कमिटमेंट कर ली। उस वक्त तो वो वासिम रूम थे, वो सबसे पहले उन्होंने मीटिंग बुलाई जी, कांग्रेचुलेशन जी, वो हमें डॉक्टर साई साहब आ गए सारे के 1.5 अरब का हमें वो सेंटर मिल गया। मैं उस वक्त कहता था ये फंस गए हम लेके ना कि मुबारक मुबारक मुबारक। अब 2-3 साल बाद वो कहेंगे जी बताएं क्या किया। तो अब जो है बायोटेक्नोलॉजी वाले जो हैं, तो वो हमारी भी फ्रस्ट्रेशन बढ़नी शुरू हो गई कि जी अब 3 साल बाद तो कुछ तो दिखाएं उनको। अब स्टीयरिंग कमेटी की मीटिंग मैंने कहा जी एंड ऑफ अक्टूबर हम आपको ना कुछ जो वो लैब बन गई। तो इसी हालांकि मैंने आपको बताया जब ये वो हमें नहीं दे रहे थे, वो कहते जी निजी बढ़ा जा रहा है। तो मैंने कहा जी हमारे पास इंस्टीट्यूट है, वहां इतने पीएचडी इतने एमफिल वो आगे करवा रहे हैं। वो सेक्रेटरी जो उस वक्त का था साइंस, उसने कहा जी मैं तो नहीं जानता। मैंने कहा जी आप देख के नहीं जानते, मेरी गलती तो नहीं है, ये क्या आप नहीं जानते एरर कहाँ है, इंस्टीट्यूट कहाँ है। अब वो आप सब ने मिलके ना, अब वो सिर्फ ये नहीं कि वो जो प्रोजेक्ट डायरेक्टर या वो 4-5 लोग एसओ, तो आप जो ये सारी, एक तो बहुत बहुत शुक्रिया साहब आपका भी, आप तीनों स्पीकर, बाकी भी ये हमारे मेहमान जो है कि शेयर किया, इससे मोटिवेशन भी होती कि किस तरह से वो डॉक्टर साहब बैठे ना आईटी के डायरेक्टर, तो आईटी का इस्तेमाल किस तरह, और ये इंटीग्रेटेड अब आईटी बायोटेक्नोलॉजी और इंजीनियरिंग वाले ये सारे मिलके हमने वो करना है वो एग्रीकल्चर के लिए काम। तो वही बात कि अगर ये एग्रीकल्चर के हमारे सीनियर प्रोफेसर तो बैठे हैं, अगर ये नहीं भी चाहेंगे तो हमें फिर भी इनको करके दे, इतना मजबूर करना, इतना वो ग्रामीण के बैठे हैं कि ग्रामीण डॉक्टर साहब इनके लिए है ये सारा जो टेक्नोलॉजी बनेगी, जो वैरायटी बनेगी। एट द एंड ग्रामीण वालों ने कहना नहीं हमने इतनी बढ़ा, चलो ठीक है, हम कहेंगे कि इन्होंने बढ़ा। तो ये जिस तरह ये मशीन लर्निंग आपने देखा कितना पीछे मैथमेटिक्स, ये अभी तो चूंकि वो लेक्चर अगर 2 घंटे का होता तो ये हमें और बताते पढ़ाते और स्टैट मैथ, लेकिन सारा करके एट द एंड वो जो प्लग एंड प्ले वाला करना उनको हो जाएगी वो, लेकर उसके पीछे सारी टेक्नोलॉजी के एक्सपर्ट आएंगे तो बात बन। अब हमने कहा था ये जो सेंटर बन रहा है एक्सपोर्टेबल वो प्रोडक्ट जो आप बनाते हैं, मुझे तो वो नाम भी नहीं याद होते वो क्या क्या उसमें से करना। तो वो अगर हम कोशिश ये है कि अब कोई एक तो 3 साल में हो जाए ना कि हम कह सकें कि ये हमारा प्रोडक्ट है, ये अगर एक्सपोर्ट करेंगे तो इतने $1 बिलियन हो गए, जो भी स्टिकर दे दें उसको तो हमने ऐसे ही कहा था। तो उसके लिए ये आपने ना जो कर। दूसरा ये डॉक्टर सुजीत सिंह इन्होंने वो कहा कि जी आप नोवेलिटी देखिए, ये एक तो आपने ये सारे नॉवल जो आपकी कंट्रीब्यूशन है इन्होंने बताई अपनी, हमारे कीनोट स्पीकर जो है नई चीजें। तो नोवेलिटी के लिए पहले देखना पड़ेगा कि ये इस तरह ये साइंटिस्ट काम करे, इन्होंने कहाँ तक किया, अब हमने उसमें ऐड किया। उसके लिए रिव्यू, जिस दिन से मैं आया हूँ मैंने वो पोस्टर सेशन किया था, तो वो रिव्यू का बड़ा मसला था। ये खासकर कुछ मैथ में ऐसे थे कि उनके आवाज नहीं करते, नहीं ये आवाज नहीं है। आप एमफिल करना, पीएचडी करनी, मैं मेरा ग्रेजुएट स्टूडेंट जब मैं कनेडी यूनिवर्सिटी में, बाकियों में नहीं। तो ये डॉक्टर फारूक पाकिस्तान से था। तो ये जब हमने वो प्रपोजल डिफेंड की तो मेरे कोलैबोरेटर जो डॉक्टर शुमर यूनिवर्सिटी ऑफ फ्लोरिडा में, वो हमारे साथ अभी तक वो सुपरवाइजरी कमेटियों में है, तो उनको मैंने इनवाइट किया। वो काफी हेल्प की। प्रोजेक्ट स्टार्ट था तो कोलैबोरेटर लाता था मैं, तो एक हफ्ता हर साल हमारे साथ रहता था। बहुत अच्छा कंप्यूटर बैकग्राउंड उसमें। तो इसका डिफेंस हो गया। मैं एयरपोर्ट छोड़ने जा रहा था तो उसने कहा कि डॉक्टर जुमान यू आर लकी, यू हैव योर फर्स्ट स्टूडेंट इज वेरी गुड स्टूडेंट। तो मैंने कहा जी उसमें खास, उसने कहा जब हमने सवाल जवाब किए हैं, क्योंकि वो जिस तरह होता है कि 2-2.5 घंटे का सेशन थीसिस की तैयारी सारी, उसने कहा जो इसने हमें कॉन्फिडेंस चीजें बताई है ना, इसने लिटरेचर बहुत रिव्यू किया है। अच्छा, ये लिटरेचर रिव्यू जो है तो ये इतना इम्पोर्टेंट है, इसके बगैर हम कुछ नहीं। अब हमने वो एमफिल का, पीएचडी का जो ग्रेजुएट कोऑर्डिनेटर बनाए कि पहले दिन से, फिर वो एमफिल वालों के लिए ना, वो जो सुबह वो मौलवी साहब टीवी पे बता रहे थे कि जी माजरत क्या होती है ना आदमी का, उसने कहा जी माजरत का एक है कि जी उज़र का बताना या बहाना बनाना, तो दूसरा मानी माफी भी है। लेकिन उज़र बहाना ये था कि जी वो इतने किराया टावर पढ़ते हैं, उनके पास टाइम नहीं भाई, 3 बजे के बाद 2 बजे के बाद तो वो नजर आते नहीं। तो पेपर पढ़ने के लिए वो घर जाके पढ़ सकते हैं ना, शाम को, सुबह को, दोपहर को। तो उसमें अब हो गया, जब हमने कहा नहीं 2 साल का डाटा चाहिए, आप आहिस्ता आहिस्ता वो पेपर भी पढ़ने शुरू हो गए, कोर्स वर्क भी हो रहा, अब रेगुलर सुपरवाइजरी कमेटी की मीटिंग भी हो रही, काफी मुस्बत चीजें हुई हैं। लेकिन अप्लाइड साइड जो है ना, वो बायोकेमिस्ट्री की, वो हमें चाहिए। वो डॉक्टर साहब को यही मैं कह रहा था, मैंने कहा जी कि वो उसकी एप्लीकेशन जो है ना, ये टेक्नोलॉजी जितनी आज बताई है, तो वो और यकीन करें कि ये ना, ये नहीं कि चाहिए हम, मैंने वो अपना इंजीनियर करना बताता हूँ, मेरे टीम इंजीनियर वाली यहाँ बैठी है। तो मैंने कहा जी वो हमने वो सीनियर डॉक्टर अहमद शफीक साहब हमारे जो टीचर थे, वो मैकेनिकल के हैं, वो मशीनें सारी चलाई, तो आप क्यों नहीं सीखते? तो एक यंग हमारा फैकल्टी, उसने कहा जी मैंने 2 स्टूडेंट को दे दिए सीखने के लिए, आप स्टूडेंट चले जाएंगे फिर मशीनें पढ़ी। हालांकि आप देखें कि ये जो टेक्नोलॉजी एडवांसमेंट है, ये तो हर साल 40 साल, 50 साल, 60 साल में जाके भी वो पढ़ना पड़ेगा और सीखना पड़ेगा। कोई मुश्किल नहीं है, बस एक ना वो हेजिटेशन है और दूसरा ट्रेडिशन है कि जी सुबह आए शाम को चले गए। तो लेकिन ये बायोटेक्नोलॉजी सारी आपको ज्यादा पता, तो आपके पास इतनी अच्छी टीम है, और मेरा ख्याल कि वो आप डॉक्टर साहब कहते हैं और इंटरव्यू करें, वो भी इंशाल्लाह करेंगे, और जो है वो भी। तो आप देखें कि आपने एक वो डिग्री प्रोग्राम शुरू किया इस साल, वो कितना रश पड़ा उसमें, इतना अट्रैक्टिव आपका सब्जेक्ट भी है और इसकी आउटकम भी जबरदस्त। ये देखिए ये जो टेक्निक्स, तो वो डॉक्टर साहब तो यहाँ से भी पढ़े, हाँ जी, तो किया, तो आप भी सारे बाहर से भी होके आए। तो ये स्टूडेंट जो हैं, और ये डॉक्टर गुरजीत सिंह, ये मेरे ख्याल से IT वालों के लिए भी इसी तरह आप भी, कि आप 1 साल उनको चूड़ी सारा पढ़ाने, लेकिन एक टर्म जो है ना, वो पूरी किसी कंपनी के साथ, किसी प्राइवेट इंडस्ट्री के साथ जो आपके रेलेवेंट है, किसी लैब में, निजी न्याब जो भी है, आपके बहुत सारी, तो 1 सेमेस्टर वहां प्रैक्टिकल ट्रेनिंग पे लगाएंगे जी। तो और इस तरह से वो, अच्छा होता क्या है कि जो हमारे जो स्टूडेंट हैं या यंग फैकल्टी हैं, बाकी एक्सपीरियंस, बड़ा अच्छा कॉम्बिनेशन है, टैलेंट की कोई कमी नहीं, लेकिन डायरेक्शन सेट करनी, तो वो अब जो है ना, आपके पास इक्विपमेंट भी वहां भी आ गया, यहाँ भी आ गया, वो सेंटर सिर्फ किसी एक का नहीं है, तो लेकिन वो क्रेडिट सबको मिलना। अगर आप काम करेंगे, आपके डायरेक्टर जो है उनका नाम होगा, डायरेक्टर के पास वाइस चांसलर, डीन, सारी टीम को क्रेडिट जाएगा, और नेशनल लेवल पे भी केरल का और नाम होगा। तो ये जो स्पीकर आज कीनोट स्पीकर और उसके बाद जो है ये टेक्निकल सेशन भी है। तो ये देखें इनके पास जो काम करने वाले, क्योंकि ये हमारे इंटरनेशनल मेहमान बैठे हैं, आपको भी पता होगा कि इनके पास टाइम इतना नहीं होता। अगर ये 1 घंटा, क्योंकि ये 1 घंटे में जो बताया इसके पीछे कई सालों की मेहनत है। तो एक तो और हमारे वो कोऑर्डिनेटर होते थे प्रोफेसर भाई मुझसे वो कहते थे कि अगर ਤੁਹਾਨੂੰ ਕੋਈ ਅਸਲ ਦੀ ਇੱਕ ਗੱਲ ਦੱਸ ਦਿਆ ਸਾਰੀ ਉਮਰ ਨੂੰ ਉਸਤਾਦ ਮੰਨ ਲਓ, ਤੁਸੀਂ ਖਿਦਮਤ ਕਰੋ। ਇਹ ਸਾਨੂੰ ਅੱਜ ਇਹਨਾਂ ਨੇ ਜਿਹੜੀਆਂ ਗੱਲਾਂ ਦੱਸੀਆਂ ਇਹਦੇ ਤੇ ਅਸੀਂ ਅਮਲ ਵੀ ਕਰਾਂਗੇ ਇਨਸ਼ਾਅੱਲਾ। ਤੇ ਹੁਣ ਅਸੀਂ ਸਾਰੇ ਇਹਨਾਂ ਨਾਲ ਵਾਅਦਾ ਕਰਦੇ ਹਾਂ ਕਿ ਅਸੀਂ ਇਹਦੀ ਐਪਲੀਕੇਸ਼ਨ ਜਿਹੜੀ ਆ ਕਿਉਂਕਿ ਐਗਰੀਕਲਚਰ ਯੂਨੀਵਰਸਿਟੀ ਆ ਲਾਈਫ ਸਟਾਈਲ ਕਰਕੇ ਔਰ ਨੈਕਸਟ ਟਾਈਮ ਜਦੋਂ ਆਉਣਗੇ ਫਿਰ ਅਸੀਂ ਇਹਨਾਂ ਨੂੰ ਦੱਸਾਂਗੇ ਕਿ ਅਸੀਂ ਕੋਈ ਅਪਲਾਈ ਕਰਕੇ ਇਹਦੇ ਚ ਸਾਡੇ ਇਹ ਆਊਟਕਮ ਤੇ ਹੁਣ ਅਸੀਂ ਵਾਅਦਾ ਕਰਦੇ ਹਾਂ ਇਨਸ਼ਾਅੱਲਾ। ਤੇ ਡਾਕਟਰ ਸਾਹਿਬ ਨੇ ਕਿ ਸੁਣਾਇਆ ਇਨਸ਼ਾਅੱਲਾ ਦਾ ਤੁਹਾਨੂੰ ਦੱਸਿਆ ਸੀਗਾ। ਮੈਂ ਡਾਕਟਰ ਸਾਹਿਬ ਨੂੰ ਜਾਣਾ ਹੋ ਗਿਆ ਲੇਕਿਨ ਤੁਹਾਨੂੰ ਮਜ਼ਾ ਆਏਗਾ ਇਨਸ਼ਾਅੱਲਾ ਦਾ। ਇੰਟਰਨੈਸ਼ਨਲ ਨਿਊ ਕੈਸਲ ਪੀਐਚਡੀ ਕਰਦਾ ਸੀ, ਵਾਈਲਡ ਲਾਈਫ ਦੇ ਪ੍ਰੋਫੈਸਰ ਸੀ ਸਕਾਟਿਸ਼। ਉਹਦੇ ਪ੍ਰੋਜੈਕਟ ਮੁਖਤਲਿਫ ਮੁਲਕਾਂ 'ਚ ਉਹ ਕਹਿੰਦਾ ਮੈਂ ਪਾਕਿਸਤਾਨ ਵੀ ਗਿਆ, ਕਹਿੰਦਾ ਮੈਂ ਇਜਿਪਟ ਗਿਆ। ਮੈਂ ਉਹਨਾਂ ਨੂੰ ਕੋਈ ਕੰਮ ਡਿਸਕਸ ਕਰਨਾ ਨਾ, ਉਹਨਾਂ ਨੇ ਕਹਿਣਾ ਅਰਬੀ 'ਚ ਨਾ ਇਨਸ਼ਾਅੱਲਾ ਬੁਕਰਾ ਕਿ ਇਨਸ਼ਾਅੱਲਾ ਕੱਲ ਕਰਾਂਗੇ। ਕਹਿੰਦਾ ਪਹਿਲੇ ਤਾਂ ਮੈਨੂੰ ਮੈਂ ਸਮਝਿਆ ਕਿ ਇਨਸ਼ਾਅੱਲਾ ਕੀ ਹੋ, ਲੇਕਿਨ ਉਹ ਕਦੀ ਹੋਇਆ ਨਹੀਂ, ਕਦੀ ਨਹੀਂ ਹੋਇਆ ਸੀ। ਕਹਿੰਦਾ ਜਿਹੜਾ ਵੀ ਇਨਸ਼ਾਅੱਲਾ ਬੁਕਰਾ ਕਹਿੰਦੇ ਸੋ ਕੰਮ ਨਹੀਂ ਹੁੰਦਾ। ਤੇ ਮੈਂ ਕਿਹਾ ਚਲੋ ਠੀਕ ਆ, ਨਹੀਂ ਕਰਦੇ। ਮੈਂ 에어ਪੋਰਟ ਤੇ ਚਲਾ ਗਿਆ, ਇਜਿਪਸ਼ੀਅਨ 에ਅਰਲਾਈਨ ਸੀ। वो कहते इंशाल्लाह फ्लाइट विल बी एट दिस टाइम। कहता मैं कहीं भी नहीं कंफर्म लेकिन ये ਅਸੀਂ कंफर्म करांगे जी। कोई डॉक्टर साहब ਤੁਹਾਡੀ ਟੀਮ ਨੂੰ इंशाल्लाह लेकिन और देखो डायरेक्टर और बैठे हैं, इन्होंने और डायरेक्टर खजाने ਦੇ ਬੂਹ ਖੋਲ ਦਿੱਤੇ ਕਿ ਤੁਸੀਂ ਸੈਮੀਨਾਰ ਕਰ ਲਓ, ਤੁਸੀਂ ਬਾਹਰ ਜਾ ਕੇ ਕਰ ਲਓ, ਤੁਸੀਂ ਪੇਪਰ ਪਬਲਿਸ਼ ਕਰਨਾ, ਤੁਸੀਂ ਕਿਤਾਬ ਪਬਲਿਸ਼ ਕਰਨੀ। ਔਰ ਇਹ ਸਾਡੇ ਸੀਨੀਅਰ ਕਿਸੇ ਵਕਤ ਡਾਕਟਰ ਸਾਈਦ ਸਾਹਿਬ ਦੱਸਣਗੇ ਕਿ ਜਿਹੜੇ ਡਾਕਟਰ ਖਾਲਦ ਸਾਹਿਬ ਜਦੋਂ ਇਹਨਾਂ ਨੇ ਇਨਵੋਲਵਮੈਂਟ ਪੜ੍ਹ ਸ਼ੁਰੂ ਕੀਤਾ ਬਜਾ ਕੇ ਤੁਹਾਡੇ ਤੋਂ ਅਸੀਂ ਸਟਾਰਟ ਕੀਤਾ ਕਿ ਜੀ ਇਹ ਜਿਹੜਾ ਨਾ ਇਹਦੇ ਵਾਸਤੇ ਕੋਈ ਨੋਵਲ ਆਈਡੀਆ ਲੈ ਕੇ ਆਏ, ਕੋਈ ਇਨੋਵੇਟਿਵ ਕੰਮ ਲੈ ਕੇ ਆਏ, ਉਹਦੇ ਵਾਸਤੇ ਫੰਡਿੰਗ ਦੀ ਕੋਈ ਕਮੀ ਨਹੀਂ। और ये इंटरनेशनल सेमिनार ਅਸੀਂ कोलैबोरेशन पे बहुत जोर दे रहे हैं, इंडस्ट्री ना कोलैबोरेटर ना टीमिया ना ये ताकि सानू पता चले कि बाहर दुनिया की हो रहा ਅਸੀਂ ਕੀ ਕਰ ਰਹੇ ਤੇ ਤੁਸੀਂ ਇਸ ਤਰ੍ਹਾਂ कोलैबोरेशन ਤੁਹਾਡੀ ਕੁਝ ਵੀ इंटरनेशनली है, बाकी भी कुछ करके ये कुछ ऑर्गेनाइज कर। और जूम पे जो है वो मैंने देखा कि ये देखे कोरोना में अच्छी चीजें भी हुई कि ये जूम आया, आप तकरीबन 100 तकरीबन नहीं बल्कि पूरा 100 के करीब वो ऑनलाइन उन्होंने आपका लेक्चर सुना, उतने हाल में नहीं जितने पार्टिसिपेंट उधर हैं। तो इस सब्जेक्ट में कितनी दिलचस्पी है। तो और ये जो इंटरनेशनल स्पीकर आए हैं उनको, और वो अभी तक देखें कि वो बैठे हैं, इंटरप्शन भी हुई लेकिन वो गए नहीं। तो ये इसको हम कंटिन्यू इंशाल्लाह रखेंगे। और ये टीम जो है हमारी बायोटेक्नोलॉजी की, अब ये डॉक्टर आपदा ने ज्वाइन किया है, तो ये भी सीनियर है, तो सेंटर में इनको भी ड्यूटी दी है जो हमारा नया सेंटर है। तो लेकिन इसमें हम वो एग्रीकल्चर वाले आईटी वाले सब इंशाल्लाह जो है हमारे साथ हैं। आपको उसी किस्म की सपोर्ट चाहिए और से चाहिए और गवर्नमेंट से चाहिए, जिसका आप सारे जो है वो बड़ी हेल्पफुल है। और डॉक्टर साहिद साहब आप ज्यादा एकेडमिक्स को देख रहे हैं, तो कोई मसला हो आप डिस्कस कर सकते हैं, लेकिन ये है कि वो डेडिकेशन चाहिए और हार्ड वर्क। थैंक यू वेरी मच।
Dear listeners, it's time to present souvenirs to the university members and our respected guests. I would request Vice Chancellor Staff to present the souvenirs. So, the first name is Dr. Gurjeet Randhawa. Wasim Heather. Professor Dr. Khalid Saifullah, Director OA. Mr. Saqib Majid, Director. External Linkages. Professor Dr. Mohammad Javed Asad, Director UIB. Assistant Professor Dr. Usma Abdullah. Mrs. Burji. Vice Chancellor, Professor Dr. Kamrul Zaman. Dr. Fiola. Hamid Batu. Miss Tarandeep. Dr. Burji. Respected Vice Chancellors, guests, and committee members, who all are requested to assemble in front of the department for group photos. After that, you all are requested to move to committee room, and students are requested to move to the class for a cup of tea.